been able to. So, in an evolutionary sense, this individual currently has a fitness of 0, and there are a number of people in this room who have higher fitness than this particular swimmer. On the other hand, there's this family who I no longer wish to name specifically, but they have 19 biological children among them, leading them to be an extremely high fitness in modern society. But the problem is that these measurements of fitness are snapshots. It's entirely possible that this individual is going to have children at a later time. It's also possible that these parents are going to have fewer grandchildren than they would have had if they had a smaller number of offspring and invested more resources into each of them. So a lot of our measurements of fitness, unfortunately, are snapshots. But with the experimental systems that I work in, we can actually integrate over the entire lifetime of organisms and actually go across multiple generations to establish what the patterns are in fitness. Now, a lot of our thought about evolutionary biology is driven by this metaphor of the adaptive landscape. And this was derived by Sewell Wright back in 1952. And there are a number of different ways that this has been set up. But the concept here is that if you imagine a simple three-dimensional landscape, two of our axes here are some sort of trait. You can think about this in terms of the specific genotype, so the specific DNA bases that they have at particular locations, or you can think about this in terms of traits such as body size or metabolic rate or running speed. For every combination of these traits, there is some associated fitness value, which here is the height of the landscape. Individuals will exist somewhere on this landscape as a single point, and populations will exist as clouds of points. Now, some of these individuals are higher fitness than other ones are, and therefore they will have, on average, more offspring. But because those offspring are going to more closely resemble their parents, this means that the population across generations will shift upward on one particular peak, possibly until it reaches the top of that peak. Then there's a possibility it could actually get stuck here, although as we will see, some of our intuitions about how geometry works in 3 dimensions may not actually be applicable to the much higher dimensional surfaces that are involved in real adaptive instincts. Now it turns out that cloned Earths are difficult. And despite the fact that we have a tremendous amount of funding in our lab, the NSF has not allowed us to actually set up multiple independent Earths. But we can treat different flasks as different worlds, because each of these flasks can evolve independently of all of the others. So most of my work for my dissertation has been in this particular experiment in Richland Seedlot, the long-term evolution experiment. This is a very simple experiment, starting out with 12 populations of E. coli, all derived from a single common ancestor, and all evolving independently of each other. Every day, someone in the lab goes through and transfers each of these 12 populations, taking 1% of the liquid that's in there and putting them into a fresh flask with new sterile growth. Our growth media is basically just a salty sugar water, and the sugar is what's controlling how much they can grow. Because we do a 100-fold dilution every day, and they grow back up to the carrying capacity of the system, there's a 100-fold growth every day. And that means we get just about 6 2/3 generations, which we call close enough for rounding error. So every 75 days is 500 generations in the system. This has now been going on for more than 60,000 generations, although the data I will be talking to you about today comes only from the first 50,000 generations of the experiment. The great thing is, because these are frozen every 500 generations, we have the entire history of what is going on in these populations. And if I want to compare populations from now to populations from 20 years ago, I can pull them both out of the freezer, grow them up in the lab at the same point, and compete them directly, rather than having to rely on data that someone else collected 20 years ago. Now, for 50,000 generations, this is a gigantic amount of time. It's hard to keep in perspective exactly what that means. So a couple benchmarks I like to use. Corn was domesticated within the past 10,000 years, and since it's an annual plant, we go through about 1 generation a year. So in about 10,000 generations, we went from Teosinte over here, which is the wild relative that corn was derived from, to a year of modern corn. This one in the middle is actually an F1 hybrid, so one of its parents was a Teosinte, the other was a modern corn. This is a tremendous amount of differentiation, but this is all driven by artificial selection. Humans actively choosing which organisms they're going to use to produce the next generation. And there's a possibility that artificial selection could allow things to happen a little more rapidly than you have in natural. So to get at that, we can put things in perspective of hominids. So we have a Neanderthal on one side here, and an anatomically modern man on the other. Our two species diverged from each other about 30,000 generations ago. We had coexistence about 2,300 generations ago. This is still only a fraction of the evolutionary time that we're talking about in the experiment. Now, to get at all of these things, we need to actually be able to quantify fitness. And so as Rich alluded to, I spent years of my life counting colored dots. And the basic principles of this are fairly simple. We go into our freezer and we grow up our two different populations. One of them is whatever one we want to count fitness for. The other one is just a reference strain that we can compare. We grow them up, transfer them into the media in which they've been evolving, which is the same media that they're going to be competing in, just to get them physiologically acclimated to those growth conditions. And then take a sample from each one of them, mix them together, and immediately run a dilution series on a small portion of that liquid. We put them onto a plate where we can tell the difference between the two different colony types. We let the rest of the competition grow in the incubator for 24 hours, and then repeat that. And by looking at these colony counts at the end of each of these two growth cycles, we can determine what the population size of each of them was. Now, these are showing everything as a one-day competition. Sometimes when I need a little more precision, I can run things out for longer and take into account the additional generations that this competition occurs. So data I will not be talking about today. In chapter 1, we tried various different methods of doing fitness. We found that the one we've been using forever is just as good as everything else we tried. So it's a good reason to continue using it. But now going on to some of the actual fitness results. One of the questions we had is fitness still increasing in the system? This is a very simple system where we don't have a lot of the things that we know slow down adaptation within natural populations. And so it was interesting to us to figure out whether fitness was actually continuing to go up. So here I'm going to be plotting fitness at 40,000 generations on the x-axis, fitness at 50,000 generations on the y-axis. This line here is just the y equals x line. So anything that's up here is more fit later on. Anything that's down here is more fit earlier. When we plot our populations on there, this is what we get. All error bars I will be showing throughout today are 95% confidence intervals. What you can see is above and to the left of the line, showing that fitness has in fact increased over these final 10,000 generations that we're talking about. We can also do stats on that and show you that it's actually a significant result, but the data should be clear enough in the picture that you can see that these populations are in fact continuing to adapt even over long periods of time. So now that we've established that, we need to figure out how that is happening. So as Rich alluded to before, we have our 12 populations. I took 41 time points from each of the 12 populations done in replicate to figure out what was happening on average to populations in our experiment. And this is the basic result there, and for some reason, my PowerPoint just crashed, and so let's try that again. So what we find out is that we have a general trend of how fitness is changing within our system. And it turns out that you have these relatively rapid gains early on, followed by a deceleration in how rapidly fitness changes. There we go. And so we have this general shape, and we'd like to figure out how we can describe that mathematically. So one of the equations that people have used traditionally in our lab is this one here, which is a hyperbolic fitness function. This is a fairly simple function. It just has two parameters here, a and b. T here is representing time, and w is our standard notation in evolutionary biology for fitness. And this has this basic shape here. Every time I'm going to plot a red curve, it's going to be this hyperbolic function. You can see this does a pretty good job of going through our data, but there are some potential problems here. It tends to underestimate fitness out here at the later time points, while somewhat overestimating it early on. And we already knew that there were going to be some problems with this function, largely due to the fact that this is a bounded function. As our time value gets excessively large, there is a theoretical maximum that this equation can reach, but they can never go higher than that. And when we fit these equations into the path, we've noticed that that theoretical maximum is often lower than the final fitness we actually calculate after we've done so. So we know there are some problems with that. So we decided to try fitting a different mathematical type of function. And this is known as a power law. So the big difference between these, although they have the same number of parameters in them, is that this is an unbounded function. This will continue to decelerate as time goes on, because our exponent here is a positive value, but it is less than 1. So if this was equal to 1/2, this would just be a square root function. So as time goes on, this will decelerate and grow slower and slower, but it doesn't have a theoretical maximum the way that the hyperbolic function does. You can see that this does a much better job of fitting our data. If we put them on the same axes as each other, you can see that this is true both here at the later time point, but also here at various different earlier time points where it does a much better job of going through the cloud of our data. Now, if you want to look at this beyond just visually, we can use statistics on it. Because these are not simple nested functions, we can't just do a likelihood ratio test, but we can look at something referred to as the Bayesian information criteria. In general, differences in BIC values between models are considered substantial if there are 2. They're considered extraordinarily strong support for one value is a 10 or greater. Our difference here is more than 3. So this is overwhelmingly more likely that this data would have been generated by a model from the blue curve than if it was a model from the red curve. You can also look at the fact that although these are both doing a good job of describing the existing data, they do a very different job of predicting future data. So if I look at this hyperbolic function, and I fit the curve for just the first 10,000 generations, 5,000 generations in this case, and project what that curve says fitness will be over a long period of time, I can then add in more data. And every time I add in more data, you see that this curve shifts. And it's always shifting upward because we're always under-predicting what the later fitness will be. So even though this particular function does a good job of describing whatever data we use to fit it, it does a terrible job of predicting the future. On the other hand, from the power law, as I add in more data, my curve basically doesn't change. And so I can use early data to very accurately predict much longer period trends, which makes it much more likely that this is describing how fitness is actually changing within our environment. Another way of visualizing the same thing is here if I look at just the first 20,000 generations and fit both of the curves. I project what each of those curves say fitness will be over long periods of time, and then I add in the data that I wasn't using to fit the curve. And you can see that this blue curve goes right through the middle of these points. So this is telling us that we can actually predict fitness over long periods of time in this system. And the great things about it is this also tells us that fitness is increasing indefinitely. Our previous discussion about adaptive landscapes, most people tend to assume that populations out in nature are staying at or very near the top of the fitness peak unless the environment has been very recently disturbed, most likely by humans. But these are populations that have 50,000 generations to adapt to a basically unchanging environment, without predators, without prey, without any other co-evolutionary pressures. And they are so far from reaching a fitness peak that we can't detect evidence that a peak even exists. So maybe it's not reasonable for us to assume in all of our ecological models that populations out in nature are already as adapted to the system as they are likely to get. Predictability. But then we'd like to look at repeatability. As we had said, there are in fact 12 different populations within this experiment. So you can fit these basic curves for all of them. And I don't expect you to be able to read all of these things in the back. But I can tell you that when we fit these curves for all of our 12 populations, in 7 out of the 12 cases, one of the models is in fact substantially better than the other. And every single time that's true, it's the power law is better. Out of 10 of the 12, the point estimate says that the power law is better. But in a couple of those cases, and in all of the cases where it looks like maybe the hyperbolic is slightly better, it's not a meaningful difference. And so it's just noise. This is showing us that our power law trajectory is not just an artifact of looking at the averaging across many different replicas, but it's actually what's going on within each individual population too. Now at this point, everything I've presented is purely empirical statistical results. And I gave one of these presentations a number of years ago when we had Noah Rybeck, who was a new, at the time postdoc in the lab who saw me give this presentation and decided that he wanted to derive a population genetics model to figure out how fitness should be changing in our system. So this is a 4-parameter population genetics model using many of the standard parameters of population size, mutation rate, and distribution of beneficial fitness effects. And he ended up with the same functional form of the power law I did. We were fully expecting my statistics to simply be a gross generalization of a much more complicated formula, but we end up with the same mathematical function. And because this is a population genetics model, it has parameters that we know have changed within a number of our populations. For instance, about half of our populations become hypermutators, increasing their mutation rate somewhere in the neighborhood of 100-fold over the baseline mutation rate. And 3 of them happen to happen relatively early. So we decided, well, we can use Noah's model, fit what things are going for the ancestral population, and then change the mutation rate for the hypermutators at the point they become hypermutators. And we get this. The curves here show what the model is predicting. The symbols show the actual data for the different populations. So we fit this black curve on all of these dots. Then we knew that this green curve were the things with high mutation rate. We changed the mutation rate in the model at this point and got this curve, and then plotted the data on top of it. So that curve was fit without all of those green points of data, and it happens to describe exactly what we're seeing. The fact that we have a theoretical model for this, in addition to purely statistical empirical results, shows that this is very likely what is actually going on in our system. And so from our bacterial data, we can show that fitness changes are predictable. We can use just the first 40% of our experiment to predict how fitness is going to change far into the future. And our fitness changes are repeatable, but we get the same basic patterns across many different variables. It's also true that our predictions are relatively reasonable. So we did some back-of-the-envelope calculations and said, okay, 50,000 generations was approximately 1 scientist generation doing this experiment. What if we go out 50,000 scientist generations of this? And we assume that all of the increase in fitness comes at decreasing the doubling time. And we find that this gives us a totally reasonable value, somewhere in the neighborhood of 12 minutes of our doubling time. We also ended up writing this out even to at the point when the Earth is consumed by the expanding sun. And we get generation times that are within the realm of what we have seen in other bacterial species in somewhat different rhythm. Now this is a slightly harsher environment in that we don't give them all of the components, they have to make some things themselves, but it's not like we're predicting that they're going to start doubling every 30 seconds. These are actually reasonable values. So the question that I had left from the beginning is whether this is a general phenomenon. And for that we decided to look at a very different system. Which is ADIDO. ADIDO, for those of you who aren't familiar with it, is the digital evolution software platform. Organisms within this world are self-replicating computer code, but the user can define mutation rate such that not all of those copies are perfect copies of the parent. The programs are always competing for space within the world, and depending on the environment the user finds, they may also be competing for CPU cycles to replicate their code more quickly and therefore be more likely to have offspring. Therefore, these are things that exhibit inheritance, selection, variation, and they have generational time, which means that this is an instance of evolution rather than just a simulation of it. This screen grab happens to be from ViaEd, which is an educational version of this software, developed here with a graphical user interface. And research actually I've done takes place with the command line version, which doesn't use itself to visualize. For the experimental design, we decided to look at 3 different environments. One, a very complex environment with a very large number of tasks that organisms could evolve to perform. One, with a very simple environment where all they can compete for is space within the world. And so they're only trying to become more efficient basal replicators. And one that's sort of intermediate, that is the most previously studied of the environments within the system. Where there are only 9 logical tasks that you can do, but they get varying different degrees of reward for doing simple ones versus complex ones. We ran these out for a very long period of time. In the logic environments, we ran these for 200,000 generations. For the no-task environment, we ran them out for 1 million generations. One of the great things about working with computers is they go even faster than the bacteria. We can look over very long periods of time. We have 20 replicates of each one of these things, which thankfully the computer does all the math for me, and I don't have to sit there and actually count the number of organisms of each type, because that would be a little tedious. So we're going to step through the results, and for some reason, PowerPoint has decided to crash again. I have no idea what's going on, but let's relaunch that. So the first environment that I'm going to be talking to you about is the Logic 77 environment. This is the most complicated of the environments. There are 77 different logical tasks that the organisms can perform. And any one of the tasks gives the same degree of reward as any of the other ones. So it doesn't matter how complex the task was. You're rewarded simply for doing the task. In some ways, this is the most biologically relevant of the different environments we're talking about, because there are lots and lots of different things that cells can get better at. They can get better at internal enzymatic reactions, they can get better at withstanding different environmental stresses or picking up different resources. So in that way, this is a little more representative. Now, this is the sort of curve we see in fitness within development time. But again, we have this characteristic shape of a rapid increase followed by an acceleration. Note for those who are a little more math-minded in the audience, this is actually a log scale on the y-axis, so things are increasing much, much more than we were seeing within the background. And the thing is, when a number of people have done AVITA runs in the past, they run them for a certain length of time, and they see that fitness is leveling off, and so they decide that the populations have basically finished adapting to that. And that is not necessarily the case. What we see as fitness plateaus are actually just artifacts of sampling. So if I take this same set of runs and look just at the early data, I see that this is what it looks like over the first 2,000 generations. So you see that it increases and then it kind of levels off. But as I zoom out, and every time I zoom out, the end of the previous graph is going to be a dashed vertical green line. You can see that we keep getting the same characteristic shape. And it keeps looking like things have leveled out. But as you zoom out, what looked like a plateau before is now a steep portion of the early part of the curve. And so just because something looks like it's leveled out doesn't mean that it necessarily has. Now from this data, we can fit the same sorts of models that we were fitting before. We can look at what a hyperbolic fit looks like, and you can see this actually does a very good job of explaining what's going on in the system. But the power law is substantially better. If you put these on the same axis, you can see that the blue line all but disappears into the black line, whereas this red line is showing deviation from it standing out. As I said before, we have overwhelming support that our bacterial data was better fit by this power law than Herbola, because our difference in BIC value is about 34. Our difference here is more than 10,000. So this means that the ratio of the probability of getting this data from the blue model versus the red one is more than 10 to the 2,000. These are just ludicrous numbers that are almost meaningless to humans. We again see that the power law also does a better job of predicting data. So just as before, I use the first 40% of the data, fit both of the models, predict what they say fitness is going to do over long periods of time, and then overlay the actual data. And in this case, it turns out that the power law is slightly overestimating it, but it's also doing a much closer job of predicting what that data is going to do than this hyperbolic function, meaning that it looks like fitness is not bounded function within this system either. Now it turns out that the different replicates within the digital environment are actually very, very different from each other. And so if we graph each of the population, each one of these lines here is one of the 20 different replicates, and this black overlaid line is the average across all of them. And you can see that we get very different results. Some of them are barely moving after an early time point and show that it's relatively low fitness, whereas other ones come up much higher. But even these ones that all come up very high get there at very different times. This one comes up very quickly and then levels out, whereas some of these other ones look like they've leveled off for a long period of time and then suddenly show jumps later on. So just because a particular population has leveled out for a while doesn't mean that it's not going to continue increasing in fitness later on if you give it enough evolutionary power. We can show that fitness is still increasing within the system just like before, looking at what fitness is at the end of the run versus 2/3 of the way through, we have this y equals x line. The individual points are now above and to the left. Now, because this is computational data and not us measuring things in the wet lab, we don't actually have measurement error associated with each of those fitness values. We don't have confidence intervals on it, but we can still do the stats, and yes, in fact, things are more fit at the end than they are 2/3 of the way through. The thing is, this is true across the other environments that we look at as well. Shifting to the environment in which there are no tasks to do, and so populations are simply becoming more efficient at basal replication, we get the same basic pattern. That as we add data, we crash my system again. I think the problem is that my PowerPoint is kind of absurdly gigantic because all of those points are being set up by individual replicates, and so lots and lots of points. When you have a point every 20 generations for 1,000,000 generations, it gets to be a relatively large file, unfortunately. But we get the same basic pattern here, that as we add more data into it, what looked like we had reached a fitness plateau turns out to not actually be a fitness plateau. So I'm trying to find the right slide, and it's spinning like that, which is always a little bit dangerous.
I'm going to quickly delete the first half of your talk.
It turns out that it's the second half of my talk that is much more of the data, and so that's probably what's driving it. It turns out that when you generate hundreds and hundreds of gigs of data because you run things for very long periods of time, things eventually get a little slow. I'm going to assume at the moment that you guys will believe me that as I add in more data, I continue to see this same sort of pattern, and I will move on to comparing the model fits. So just as before, we can fit the same two basic models, and this shows me that it's but this is what this looks like over long periods of time in this environment. Notice that in this one, we don't have to log scale the fitness because things are much closer to what the ancestor is. Again, we can fit the hyperbolic model for this, and it is in the process of loading down. But this does a fairly good job of predicting fitness over a long period of time. But again, the power law is actually better at doing this. And so we're going to have this blue curve when it finally shows being a little closer to that black line. It becomes relatively clear that one of them is in fact doing better. And the stats become even more absurd than I had it. If my system would allow me to continue advancing things properly, I can show you that things are slightly different actually in the logic environment. And what's different is the distribution of mutation effect sizes. Every particular task an individual gains within the logic 77 environment doubles their fitness. But within the logic 9, things are arranged from simple tasks which double fitness to a very complex task which multiplies fitness by a factor of 32. So you can get an individual mutation that multiplies your fitness by a factor of 32, and you get huge gains over a very short period of time. So it looks like in that system, what's going on is that gain of the task is dominating how fitness changes. And although there are still the fitness benefits of having lots and lots of small mutations that make things slightly better at basal replication, they end up being swamped out by these huge fitness gains from individual tasks being involved. In that case, the overall system looks a little bit more like a hyperbolic fit. But we think that this is all driven by these huge task gains. And so one of the extensions that we're doing on this actually involves using organisms that have been adapted for a long period of time within a particular environment, and using them as the seed organisms for the next generation. I'm going to try skipping ahead because I don't want this to crush on me.
That's a good idea. Yeah. We will.
I can show you more graphs if you're actually interested. But the basic take-home message from this is that in these Avita environments, we again have both predictability and repeatability of the evolutionary process. And we get that fitness is still continuing to increase over these vastly long periods of time, which even in these very simple systems, we might expect fact things would plateau, but that doesn't mean they necessarily have. The spectrum of mutational effects does have a strong effect. When you have these giant fitness things available, it's possible for things to look like they might have worked out. But even in this one environment where the whole sum of populations looks like it's leveling out, if we look at just the last third of the data, we show a significant positive increase in fitness. They are continuing to get these small effect mutations that are making them more efficient replicators. It's just the signal of that is being swamped out by the huge gains we're making really fast. So we think that this general system of power law dynamics and fitness may be a general phenomenon across the evolving system. And as I alluded to before, we think what is going on is due to the fact that this final organism is massively more fit than the ancestor by a much larger extent than anything we saw in data. So in collaboration with a number of other people, we are in the process of taking evolved organisms and using them to seed new evolutionary drugs, and then measuring how fitness changes over long periods of time in those circumstances, which is much closer to what we have in the bacterial example, because the bacteria we started with was already pretty good at growing in the media that we were using. With that, I'd like to take a few minutes to go through acknowledgements. Acknowledgements tend to be a little bit longer than a defense than from other talks, and I hope you'll do it. So the logical place, of course, to start is Rich. This is his Twitter picture. First, we met him at a conference the year before I was applying. When I was in Brendan's lab, I had no idea I was going to be moving elsewhere. Although I had been working in labs for several years, I at that point actually had no publications. And Rich just decided to take a chance on me. I actually came here thinking I was going to work on an entirely different project, ran into some methodological problems, moved on to other things. But there are a number of things that are great about working in Rich's lab, and one of them really is that students have a tremendous flexibility in what they choose to study. As long as it has to do with ecology or evolution and uses microbes in some way, it is potentially a fair game. I have the rest of my committee at the moment. Charles Ofria is a professor in computer science with whom I've been doing all of the video work. He has graciously allowed me to attend these lab meetings for the majority of my time here, including time through weekend, which has given me much more experience about talking about my work to people outside of my specific site. Tom Schmidt, who is joining us remotely, is actually the first of the MSU researchers I spoke science with. Back when I was in Brendan's lab, I came up with an idea for a particular project, pitched it to Brendan, and Brendan told me, well, if anyone who's currently working on that, it would be someone in Tom's lab, so you should write to him and ask him. And so I wrote an email to Tom, explained my idea. He said no one was doing that, but it sounded great. He had some ideas of molecular controls I could do to help ensure that what I was trying to find was something I could actually demonstrate. He allowed me to go crash his course at Woods Hole for a couple of days when I did get here in order to run some of those controls using machinery I didn't have access to. Arne Hintz was very nice to step in and be a committee member of mine for the past several months. As it turns out, if you're at a place long enough, your committee changes over time. So when I first got here, I had Andrew McAdams on my committee, a statistician in the department who decided to move back to Canada after I did my qualifying exams. So I replaced him with Ian Corkin, a statistician in the department who moved back to Canada recently. Thankfully, Arne is not Canadian, so I'm unlikely to chase him away to the Great White North just by allowing him on my committee. So I worked, when I was at Stanford, primarily with Brendan Bohannon, also some with Dimitri Petrov, working on the project Reese. And actually, my experience in working in wet labs and working in evolution in general all dates back to Steve Finkel, who was my advisor when I was an undergraduate. Being the sort of person I am, when it came time for me to do undergraduate research, I read through all of the faculty profiles in the department, ranked them by my interest, and wrote emails to the top 5 of interest to me. Steve was the only one who replied to that. And he didn't actually have space for me in his lab, but he said in good conscience he couldn't tell me no, so decided to allow me to work in his lab starting that semester even without space. And I worked with him for the next year and a half and got myself into the fun of working with organisms that evolve under timescales that I can actually trust. We have a tremendous number of people that I have overlapped with in my time here in the Webb Lonsky lab and the Vivo lab. I am just listing here grad students and postdocs, with the exception of 2 of the particular undergrads in Rich's lab who I worked with more closely. Part of the advantage of working in these environments is just how collaborative things are. What became the basis of my dissertation started out actually as a collaborative effort across multiple people in the lab. Everyone else ran into time crunch issues working on other projects, and so I ended up taking it over as the only person who had time left because I had methodological problems on a different project. The people highlighted here are people who have collaborated with me on the particular topics I was talking about today. Noah for most of the population genetics theory work. Dave Bryson was a student of Charles's who ran the initial very long runs of the data and then wanted to know what to do with the data. And then more recently, Anya and Emily are current students of Charles's who I'm working with extensions on that same thing and actually helped me get these runs to recreate things from Dave's files for which nothing was saved from. Also in particular, I do need to point out Yuja Hajima, the lab manager in Rich's lab who has helped me certified in glassware. I go through ludicrous amounts of that, and so it's really helpful having someone corralling the undergrads and making sure that all of the materials are done. The people here who are highlighted are people who I have collaborated with on other projects, in addition to the giant all-hands project, which then swamps out virtually all of the Linsky lab. These are all people I've collaborated with projects on in my time here or I'm currently doing. There are a number of funding sources I need to thank. So Beacon has supported me for some time. Michigan State University, which had a Y the last time I saved this, but apparently it doesn't anymore. I am now in the Department of Biology, which was formerly zoology until this summer. I received money from the National Science Foundation. I was actually supported for a while on one of Rich's grants from DARPA, which I find bizarre that I was somehow involved in DARPA. And ICER, who is our Institute for Cybersecurity Research, for the computational support for the high performance computing cluster that has given us tremendous ability to run things. And beyond that, there are also lots of people. So the lab has changed sometime over the years, the Linsky lab in various different iterations, the Devo lab in various different iterations, all people I've been discussing about in research. I also have friends from various different sources that we have gone bowling, or done curling, or played laser tag, or silly cats at a wedding. As many of you may notice, I have spent some time playing complicated board games down here, or in more recent years. Oh, I lost that picture. I also have several pictures of the fun of complicated pumpkin carvings. Grad school can be an isolating place if you don't have friends available, and this is to what degree I can approximate sanity. There is also my family, my father, who is was unable to make it. He's actually a game theorist, currently in economics, formerly in mathematics. It's fun coming from a family of nerds, where no one really had a problem with the fact that their science-minded son didn't feel the need to go on in a position. And with that, I'd be happy to take some questions.
Really clear presentation and being able to talk through technical difficulties makes it even more amazing. So the kind of convention, for those of you who don't know it, is that we have questions from everybody except the committee, and so Mike, you can take those, starting with Tom.
Mike, if your PowerPoint is 365, at any rate, if it will allow me, mind going back to the slide where you first hit the power law to the 2000.
To the bacterial data?
Yeah, bacteria
Yep.
data.
Yep.
While you're on there, I'm completely won over to the power law. I can't argue with BICs like that. But when I look at that graph, it looks to my eye, which can be deceiving, it looks like the deviations from the model are autocorrelated. You have runs below the line, and then you have runs above the line. And statisticians might get in a twitch about that. I don't care. But I'm wondering if there's some biology story that's how it is that you get a long run above the line, and then it's almost like it's homeostatically dropping down and cycling above and below the line.
Well, so one possible explanation for why we have these runs that are a little bit below the line for a while is because of the fact each one of our 12 populations, we took the various different replicates, we averaged that to get what that population's score is at that particular time. So if we have deviant populations, one that is, say, much higher than average or a couple that are much lower than average, you can have the outliers pulling the power law calculation slightly out of whack to what it's doing to what you see as the mean. So the overall answer to that is that individual populations can have differential effect on how we fit the equation versus what the mean looks like. And that can cause these runs because those would be autocorrelated in that a population that has accelerated in fitness compared to others will have done so for some length of time, in the same way that one that was lagging behind will have done so for some length of time. Josh?
Yeah, so the experiments both in Amoeba and in bacteria have been done in very, very constant environments where everything's possible to try to keep conditions the same. And you see this decelerating improvements. So I would imagine in the natural world that at some point those other factors start to come in. Mild changes in the environment over the course of tens or hundreds of thousands of generations. So do you think that that limits the applicability?
So expect things to be improving more continually than we have in this very constant environment, where there is potentially a theoretical best value that they could be getting to. Therefore, out in nature, we would expect fitness plateaus to be even harder to reach than they are within the laboratory environment. And within the laboratory environment, we don't see populations reaching fitness plateaus even over vastly long timescales. It is unlikely that many populations out in nature will be experiencing an unchanging environment. Therefore, I think if anything, that further strengthens the idea that populations in nature are probably not actually reaching fitness plateaus. Jason?
Since we got this up here, so the other thing that struck me with this is your variation is larger each over time.
Yes, yes it does.
Do you think that's just a statistical thing, like the bigger mean, the bigger, or is it actually real? Like Gavlet's idea of holy landscape is that there's once you get high enough dimensional space, you have all these different ways of having essentially the same fitness. Is that what's going on here? They're just kind of dancing around? Or?
So the basic question here is based on the fact that these error bars here tend to be a little bit smaller than these error bars out here. And there are two different reasons for this. One of them is actually methodology. So the particular way in which we happen to assay fitness is more sensitive when values are very close to 1. So as you get out further, that variation becomes higher because things are rising in fitness. This was one of the reasons why we had tried several other methods to calculate fitness. We get the same we get almost exactly the same fitness values. We get slightly different coefficients of variation. Sometimes one method is a little bit better than another, but that changes over time. There's also the fact that there is actually population differentiation. So for instance, we have one population that is lagging well behind the others in fitness. And so it's staying relatively low over long periods of time. So even when all of the other ones are going up, that means that that one, which is basically flat, is dragging the error bars to be larger and larger because there is more differentiation among different populations in these later evolutionary time points than there are early on. Alita?
So given that we have only one Earth, are there any trends that we can see with these diminishing returns patterns in the fossil records? If you have a nutrient evolve that increases rapidly at first and then plateaus out, or in terms of fitness?
So I have not looked at fossil data, in large part because I can't really correlate fitness all that well to the sorts of traits that we can fossilize. A particular organism or a particular fossil being more or less abundant may be related to being more fit and therefore there being many more of them in the population. But it could also be due to them colonizing a particular environment that was particularly likely to lead to fossils. the sort of measurements of fitness that I'm doing are relative fitness showing the likely population, and that sort of data is not necessarily fossilized well, so I don't think I'd actually be able to look at it that way.
How about for traits, say, on an origin of an adaptive radiation, you might see a trait shoot off in one direction quite rapidly at first, and then it reaches
Okay.
or deceleration.
So if I'm understanding you correctly, you're looking at adaptive radiations in the general model of a punctuated equilibrium, where there's like some period of time where things are relatively static, and then new things arise and change rapidly, and then things are somewhat static again. And so you get this sort of stepwise change within populations. And my understanding is that much of what is driving why we have this particular power law is that it is sort of a summation of a bunch of different stepwise changes. That each individual mutation that is beneficial within the system arises, sweeps through the population, and then is basically static. But we know that on average, each new mutation is less beneficial than the previous one benefits, which means that the step is both shorter and deeper because it takes a longer period of time for it to fix. When you sum up a bunch of things that go from being a large step that's very shallow to smaller steps that are much deeper, you'll end up getting a power law dynamic out of that over long periods of time. So if we were to see these same sort of stepwise changes in trait evolution in a punctuated equilibrium model, I would expect that we could therefore predict how those things are likely to change over very long periods of time using a power law approximation. Right.
So, in the bacteria, you saw that you saw the rise of hypermutators quite often. You actually, from the it looks like that by increasing the mutation rate there, you up the top of the predicted line. Correct? Does that make sense?
Yes.
Okay. Is there going to be a point where decreasing the mutation rate is the best way to increase the power law predicted in the future, or is it always going to be a case where having the higher mutation rate is going to be better?
It is actually the case that there are times when decreasing the mutation rate will increase the fitness of the population. When populations are waiting for the next beneficial mutation to occur, there is some benefit to having a higher mutation rate because you're more likely to reach that next beneficial mutation faster. But beneficial mutations are not the only mutations that are occurring. There are also detrimental mutations that are causing a mutational load on the population. And so we actually have evidence from at least one of the populations in Wild-Ross 2013 paper showing that one of these populations that became a hypermutator, we then had independent origin of two different clades that had mutations that reduced the overall mutation rate, but they only reduced the mutation rate if they occurred on the background that already had a higher mutation rate. And since we have two different clades doing that, it's very likely that there is actually adaptive value to it, that it's not just a one-off thing hitchhiking with something else. So we do have evidence that as you move through the evolutionary time, it changes whether it is beneficial to increase or decrease your mutation rate within the system.
Is that something you could model exactly using a population genetics? Like actually come up with what the mutation rate optimally should be at every time point?
I think it might be possible, however, I was not the one to do
Yeah,
the modeling, so I am not the best person to answer that question. But Noah, who's in the audience, is someone you can talk to about this. Cliff.
So, two questions. First, this is technical. The Avita graphs, are they average population or lands?
They are average population.
Second question. It seems that there is some principle that's existing in both the biological system studies and Avita, that there's some, at any given point, distribution of positive mutations of a certain value. I mean, I could imagine a system that had a very sort of artificial fixed genome that had within it a fixed number of equally beneficial mutations that each had a 1 in a million chance, and then you would expect this sort of linear, as it finds that, right? There's no episodic effects. And so it seemed, what's interesting is that there seems to be some sort of underlying effect on the likelihood that wheels will give you a certain benefit at a certain rate. And it seems to be behaving in both the biological and this digital system. Sort of the underlying why do these, the curves are definitely the same, but why?
So, to answer that question, I do think that the mutational spectrum is very different in these two different systems. The largest fitness benefit we know of in the bacterial example is somewhere in the order of a 13% increase in fitness as opposed to 3,200%. So there are differences in the spectrum. What I think is actually driving this evolution of the power law is the fact that our systems do have epistasis. So for those of you who are familiar, epistasis basically refers to the effect of a particular mutation depends upon the rest of the genetic background. So something that might be beneficial in one background might actually be detrimental in another. And our systems have pervasive epistasis, that mutations depend highly on what's going on other sites in the genome. So one of the extensions that Josh and I have talked about doing to this is to run this in an even simpler computational system, that of the NK landscape, where you can independently tune the amount of epistasis present within the system, and so we can see whether there's a sharp transition to things behaving in power law dynamics as we get epistasis up to a certain value, whereas beneath it we would not expect that to be true. So that's something we're going to be working on, but we have not done yet, so I have no data to actually support that. That's just a hypothesis. Bob! From that.
So if I understand right, you say that the rate of fitness increase decreases, and you're saying that it follows a power law maybe because it decays like, say, 1 over square root of x or something like that. That would give you square root function. Why doesn't it decay like 1 over the logarithm? Can you reject that using your data? Why maybe it decays really, really slowly? And you kind of answered it maybe with your sense of why there should be a power law in terms of time steps. But I wonder how hard that would be to reject that.
I have not exhaustively tested all the various different unbounded functions that are common.
Well, sure.
So it is entirely possible that there's another one which may do a better job of fitting our statistical data. But the fact that Noah's population genetics model ended up with the same functional form of the power law as I did makes me think that there's something to that particular equation rather than just any equation that isn't a bounded function. So I don't know the definitive answer. I could try other functions if there are particular other functions you would like, but I think that the general argument is that the differences we're seeing here are between bounded and unbounded functions. And the unbounded function that we happen to be using is also the one that, working from a population genetics model, we ended up with the same one. So that's a good reason, for me at least, to continue using this particular unbounded function.
All right. Again. So we'll take a few minute break and then Mike will meet with his committee. We don't know how long that goes. That's an open-ended thing, the way these things work. But we will have refreshments, including champagne, somewhere in the, I would guess, maybe an hour from now. And so
there's also a tremendous amount of food for anyone who wants it.
So we'll hunt down people who are interested in hang around or definitely out. So I hope to see some. Oh, that. Yeah, I believe it'll be vacant. Seminar room, or we'll be in the same floor talk. Yeah, I'd be willing to bet a bottle of wine without getting there is no significance in that run test. You know, that's in the run test. You know, he suggested that there were 7 points that were below the line. They're always going to be 7 points below the line, right?
And if you remember,
Actually, I think the explanation that you gave
there was
Right. We got more problems with that. like having
Yeah, well, there's
Yeah, exactly, that route, or like doing it, but.
But also, even just the measurement error, just not even